Folks, welcome back. And let me just begin by saying I hope you're doing better than I am today. I woke up with a stiff neck, having trouble turning my head that way. Boy, don't ever get old. Anyway, in this video, I want to give you some additional information about the cell cycle, which I brought up in the previous video. There are, as I mentioned in that video, there are numerous factors involved in the regulation of the cell cycle. But what I also want to point out is that mitochondrial DNA has a role in the cell cycle too. And mitochondrial DNA, the mitochondria itself, are another area where the evolutionists use their sleight of hand and try to convince us that this organelle and its DNA are part of the process of evolution. So let me address this topic for you. First of all, the evolutionists teach that the mitochondria and its DNA became part of a eukaryotic cell via the endosymbiotic theory. And just by way of reminder, eukaryotic cells are those that make up what most people refer to as higher organisms, like us, as opposed to bacterial cells, I think I said it right, which are prokaryotic cells. And eukaryotic cells and prokaryotic cells have numerous differences between them. For example, eukaryotes, like us, have a nucleus in the cell which houses the DNA, while prokaryotes, they do not. Eukaryotes have chromosomes, while prokaryotes do not. Eukaryotes have organelles, while prokaryotes typically do not. Well, with that said, there are some bacteria that have species-specific structures that are considered to be organelles, but prokaryotes don't have the classic organelles that eukaryotes have. And examples of those include the endoplasmic reticulum, or ER for short. I said Eeyore, the guy from Winnie the Pooh. that's what I meant to say. And the ER comes in both smooth and rough varieties. So I guess you could say you can have your ER with or without nuts. I thought that was funny. Maybe you will. Anyway, the rough ER is covered with ribosomes, and they translate RNA into proteins. The smooth ER functions in lipid metabolism and drug detoxification. Another organelle is the Golgi apparatus, which modifies and packages proteins, which are then distributed to their intended locations via things called secretory vesicles. So the Golgi is like the FedEx or the UPS of the cell, packaging and delivering. And then there's the mitochondria, the powerhouse of the cell. It's the site for the last two steps in the process of cellular respiration, which results in the production of ATP, adenosine triphosphate, the power currency of the cell. It's the energy source for all our cellular functions. ATP is like a little battery, stored with energy, ready to be used when the cell needs it. ATP is also one of the nucleotides for RNA, which we'll discuss further when I talk about the functions of DNA in molecular biology. So those are some examples of organelles. And now, with that said, let me get to the endosymbiotic theory. The endosymbiotic theory supposes that a eukaryotic organism engulfed an entire bacterium, a prokaryote, and this engulfed bacterium and its genome became the mitochondrial organelle containing the mitochondrial DNA. And this so-called theory is worth further discussion at this point. Mitochondrial DNA is the additional DNA that exists in the mitochondria of our eukaryotic cells. Mitochondrial DNA is distinct from our genomic DNA, so it's not in the nucleus and it's not part of our chromosomes. It's in the mitochondria organelle. It's also a circular molecule as opposed to our chromosomes being linear molecules. It also contains much less DNA than the genome. In humans, for example, the mitochondrial DNA spans only 16,569 base pairs. But our genome, which is all our chromosomes, contains 3.2 billion base pairs of DNA divided into 23 chromosomes, which we have in pairs in all our cells except for our gametes. So it's 3.2 billion base pairs in our genomic DNA as opposed to only about 16,000 base pairs in the mitochondrial DNA. In humans, there can be 1 to several thousand copies of mitochondrial DNA in a cell. Human mitochondrial DNA encodes for 13 proteins and 24 RNA molecules that are used to convert the chemical energy from food into a form that cells can use, namely the ATP molecule. And with that explanation, let me point out how we have a problem here. First of all, there are different families of enzymes called DNA polymerase that are used for the replication of DNA or making more copies of our DNA. And I'm going to discuss DNA replication further in a future video. But for now, I want to explain to you that the DNA polymerase that replicates mitochondrial DNA is called DNA polymerase gamma. DNA polymerase gamma is an enzyme that's composed of 3 subunits: a catalytic subunit encoded by the POLG1 gene, and a dimeric subunit encoded by the POLG2 gene. The DNA for the POLG1 gene has been mapped to human chromosome 15, while the DNA for the POLG2 gene has been mapped to the human chromosome 17. So with that said, do you detect a problem here for evolution? If the mitochondrial DNA came from bacteria, why are the genes for the DNA polymerase used to replicate the mitochondrial DNA found within the human genome? You must understand that the DNA polymerase used to replicate mitochondrial DNA, the DNA polymerase gamma, is different from the DNA polymerase used to replicate genomic DNA, which is namely DNA polymerase alpha, delta, and epsilon, and different still from the DNA polymerase used to replicate bacterial DNA, such as DNA polymerase III. If mitochondrial DNA came from bacteria, the DNA polymerase used to replicate it would be the same as that as bacteria. Getting mitochondrial DNA from bacteria without the DNA polymerase to replicate it is useless. So what was the origin of the genes that encode for DNA polymerase gamma? How could the human genome know to account for this need of this other DNA polymerase before having the mitochondrial DNA? How could that have happened randomly? Why would genes necessary for replication of mitochondrial DNA, an absolute requirement for energy for our cells, and therefore an absolute requirement for life, be housed elsewhere if it all supposedly came from bacteria? Furthermore, how could a bacterium have a genome that consisted of only the genes encoding RNA and proteins for the generation of cellular energy? What about the genes required for encoding the proteins necessary for the structure of the mitochondria itself? And to give you an idea of how many genes that would be, between 1,000 and 1,500 distinct proteins have been identified in the structure of the mitochondria of mammals. Moreover, the large number of genes that encode the structural proteins of the mitochondria are also found in the genomic DNA, just like the genes for the mitochondrial DNA polymerase. So once again, we have the same problem I already pointed out with the mitochondrial DNA polymerase genes. How could and why would an engulfed bacterium insert all these mitochondrial structural genes and mitochondrial DNA polymerase genes into multiple locations across the genomic DNA and yet keep the mitochondrial DNA separate and intact? Either this must have somehow happened, or the genomic DNA contained the genes for mitochondrial structure and the mitochondrial DNA polymerase enzymes before these structures and DNA even existed. And then there's the obvious. What about all the genes required for the rest of the bacteria? A free-living bacterium absolutely would have required the genes to encode the proteins that create the rest of the bacterium's cellular features, which would be a rather large number of genes. For example, E. coli, a common bacterium, contains about 4,000 genes in its genome. All of them are required for E. coli to exist, so that's a far cry from suggesting that a bacterium could have existed on only 37 genes, all of which are focused on energy creation. This falls way short of the other required genes for the survival of a bacterium. Do you see the mounting problem that we have here for evolution? And it also must be noted that no bacteria have mitochondria or mitochondrial DNA. Bacteria do not have or utilize mitochondria, its DNA, or the genes it contains. They utilize a different mechanism for energy generation. But supposedly, in the past, some did. Where's the proof or data to support this claim? It's simply not observed in life, and it actually makes no sense whatsoever. In addition, mitochondria are almost always received via maternal inheritance. Although paternal inheritance does exist in some mussels and insects, like fruit flies, honeybees, and cicadas, and as well as some rare instances in mice and sheep and cloned cattle. So if mitochondria and mitochondrial DNA primarily come from the mother, and if they were supposedly engulfed by a eukaryotic cell a long time ago, how did that first life form then divide its heritability in this manner? For example, if the first life form that engulfed this bacterium reproduced asexually, how was it distributed in this manner? If the first life form that supposedly engulfed this bacterium reproduced sexually, how did the males survive without it? Once again, we have yet another essential case of irreducible complexity with mitochondria and mitochondrial DNA. Therefore, you must know the requirements and specific functions of the mitochondria, the biochemistry of cellular energy production, all the proteins required in the structure of the mitochondria and their necessary genes within the genome, the DNA and genes required in the mitochondrial DNA, and the proteins and RNA molecules it encodes, the requirements and structure of the specific DNA polymerase that replicates the mitochondrial DNA, and the pattern of heritability of the mitochondrial DNA. All those things you must know ahead of time in order to plan to strategically, intentionally, and intelligently create the structure and the function of mitochondria and mitochondrial DNA exactly as required in the design of the essential mitochondrial DNA function of energy production. As I hope you can now understand, this argument and the endosymbiotic theory are completely illogical, as is the entire case for evolution. And I'm going to end this video here. But come on back for the next one, where I'm going to begin discussing the functions of DNA in molecular biology. And if you like what you hear, give me a thumbs up and subscribe to my channel. Thank you very much for watching, and as always, we'll leave the light on for you.